All right, so as I was saying, those genetics problems in chapter 12 online. Okay, try to find some time between now and Monday to do those. They're in chapter 12. They're not graded. The answers are at the end, just like you saw in your lab, your lab exercises. But again, remember to be responsible with that information, right? Try to work out those genetics problems based on what we talked about in class before you look at the answers, just to see if you get them, if you get them correct. And then we will talk about them. Yes, thank you, Courtney. We will talk about them more when we get together in lab, and we will go over them. But don't wait for that time. I'd like you to work on those before that. Let me pull these up real quick forget it. I need to get into the lecture. I'll talk about that. And the important thing to be able to solve those problems is, so these are the answers that you're looking for. So if you're doing there's monohybrid ones and dihybrid ones. I've broken them up into different kinds. Some of those you haven't seen before, like X-linked. Hi, Mariana. Good morning. How are you? So if you haven't seen them, don't worry. We'll talk about them in class. But you can do the monohybrid ones, and you can do the dihybrid ones. The answers for those, the way you figure those out is you keep in mind a couple of things. That alleles that you inherit one from mom and you inherit one from dad. All right, that's a very simple concept. When it comes to numerically, the things you need to keep in mind is when organisms reproduce, and we're talking about in terms of monohybrid, one trait. If the trait, we're talking about a particular trait. If a trait only has two alleles, and that you're going to find out today, that's not necessarily the case. But if a trait has two alleles, two alternates, purple and white, like in the case of the flowers, and if you cross two heterozygotes together, my pen is being nice to me, and then, there it is, like this, like we did in class, you're going to get this ratio. This 3-to-1 phenotypic and this 1-2-to-1 genotypic. Typic. So know those ratios, because I might give you a cross like you see there between two heterozygotes and give you something like, I don't know, 700. Okay, anyway, thank you, Penn, for your contribution of 2 minutes. 710 offspring were born, right? And I might say, okay, how many of them are going to be purple if I was talking about flowers? How many of them are going to be white? How many are going to be heterozygous? How many are going to be homozygous? And I want you to know how to apply those ratios, okay? Basically, you just take that number, that 710, and multiply it by the particular ratio. If I want to know how many would be purple, I do that by 3/4, because I know 3 out of 4 are going to be purple, okay? So practice with those ratios and practice those problems, too. You also know how to do the dihybrid problems as well, so you can start working on that. But I'm going to go ahead and pick up lecture actually at, where do I want to start? Because I do want to do a little bit of overlap. I know we're further along than this, but this is an important part. So, yeah, let me start here with this idea of true breeding. You remember when Mendel went out into the garden and he picked that first purple flower up there? He knew it was true breeding, yeah? Y'all remember what true breeding means? Who does? Who remembers what true breeding is defined as? Who else is in the room in case I haven't said anything to anybody? True breeding. Give me a definition. What does that mean? If you want to, and really, if you really want to impress me, tell me the gene. Thank you, Seth. True breeding means that an organism is homozygous, and there's two choices for that, right? You can be homozygous dominant or homozygous recessive. Now, the other day why I said you'll probably find out pretty quick that I don't have to say homozygous recessive, right? Because if I say you're recessive phenotype-wise, if I say you're recessive phenotype-wise, we know you're homozygous. Okay, but that's what we mean by true breeding. So true breeding has two answers. So how did Mendel know that was a true breeding purple flower, right? Because when you look at the way the trait's inherited, that's really all we know about a purple flower by looking at it. We know that it's got at least one dominant allele. It may not be true breeding. It could be a heterozygote. Excuse me. So what we worked out in class is this idea, wait, one more slide. There we go. Of doing a test cross. They do? Okay, I'll run with that. So we're doing a test cross. So the question is stated. This is the hypothesis right up here. Is this true breeding or not? And the answers, and those are Punnett squares down below. See, the nice thing about these genetics problems, if I ask you questions on an exam like this, if you forget or what, just run down a real quick Punnett square. Look how easy that is to do. It's obvious if that's true breeding, you'll get nothing but purple flowers. It's obvious if it's not true breeding, especially when you put it into a Punnett square, it's going to be a 50-50. So it's going to be a 1-to-1 phenotypic ratio. And we say that in a monohybrid dicross. If the critter you're looking at and asking this question about, if it's not true breeding, your offspring will be half one way and half the other. Excuse me, folks. All right. Now, I did this one on the board. Hopefully, you've practiced it a couple of times as well. This is the dihybrid cross. So if you don't mind, I'd like to go over this one more time with you, right? And if you haven't drawn it before, please draw it with me this time, okay? So let me get a white board to do this on, and let me find myself where I am so I know. All right, so here's the way we're going to do this. We're going to talk about dihybrid inheritance, and in dihybrid inheritance, we're going to be talking about, two traits at the same time. So now we're not just worried about it being nothing but a purple flower. We're worried about being a purple flower, and we might also be concerned about the height of the plant. In that slide, if you look back a couple of slides, and I'm not on the slide thing right now, you'll see that one that that all the traits that Mendel looked at. He didn't just look at flowers in terms of color. He looked at the pod shape and the pod color. He looked at seed color. So we're going to go with seed color, right? Seed color and seed shape. And if you talk about seed color, be nice if my pen cooperated. But if you talk about seed color, folks, then what we're looking at is we're looking at seeds, and I'm going to have to be really abbreviative on this, so because my handwriting, I'm using the right mouse. Yellow is dominant, okay? Green is recessive. Now, if you're following along in that figure on the slide number 10, that's what I'm getting at, right? And if we're looking at seed shape, we said that round was the dominant and wrinkled. Looks like little raisins there. That was the recessive. Okay. Now, as I mentioned, you don't worry about memorizing who's dominant recessive alleles. I'm going to give that to you. All right. So let's see. What we're going to do is we're going to cross. First, we're going to cross true breeding with recessives. And you can see it in the slide. What you get is all going to be yellow and round. So that's kind of the same thing that we did when we crossed that true breeding purple flower. Remember, that's where we say, oh, it skipped a generation, right? I want to jump into the F1 cross, though. I want to jump into the table that you see down below there in that figure. Now, I said that the understanding genetics problems is to make sure that you understand it's all about probability. It's all about flipping coins. And on that screen, if you're looking at that dihybrid inheritance screen, slide 10, it's all about law of independent assortment. Now, that's not a law like they're going to give you a ticket for, but it's we call one of Mendel's laws. And Mendel's laws, it says that these gametes, these alleles can end up in gametes separate of what other alleles are doing. So in other words, it's kind of a way of saying that the yellow alleles, or the alleles for color, are not, let's say, codependent upon the ones that are shape. They don't have to go where they go. They can go on their own. So we have to count for all the ways that can happen. And you remember from from math, I said there was a way, a little mnemonic device that you use by which you figure that all out, right? Foiling. And we don't use it to expand a binomial, but we say, all right, one allele is one gamete is the first two alleles. Okay. Outer. Penny, you remember doing these, right? In class, when I said about binomial, you give me like, oh my goodness. And yet it's not. It's not about binomials. It's just about remembering the letters, isn't it? We just can sometimes borrow stuff from other disciplines. See, and then the last one. I'm only going to write again this once. You can follow along in the table and see what's happening in the last two alleles. Would look like that. It's actually supposed to be a capital Y. See, this is when my pen breaks. This is what you'll get. All right, so if we take a look and we go down below, look at the table on the right-hand side. So, all right, I'm going to go back on to the slides here. This should have a way to quickly get back into the slide radar, but I'll get you all there. There we go. All right, so if we're looking at the table on the right-hand side, in the previous slide, I FOIL it all out, and what you see across the top, and I'm sorry, the little R's in the wrong place there. Notice it's right down below, printing problem, right? That R should be up in there. Okay, so what I have across the top and down the side are the different types of gametes that each parent can produce. And I've got that from the foiling that I showed you in the previous slide, all right? And so we put those into the Punnett square, so make sure you know how to use a Punnett square, and then we count up the phenotypic ratios, right? There is a genotypic ratio. There is how many are doubly heterozygous and doubly homozygous and all that sort of stuff. But that's just really something that's something we don't really concern ourselves with. We are concerned about phenotypic. So I want to know, what are your probability of being yellow and round, yellow and green, wrinkled and yellow, wrinkled and green? And that's the 9-3-3-1 proportion. 9/16 of the offspring from a cross like this. In this case, we're talking about color and shape. It could be could be purple tall flowers and white small flowers. If we're talking about two traits, dihybrid, that's the ratio you're going to get. That's the number you want to keep in mind. Because if you're crossing like this, crossing two heterozygotes, organisms that, oh, now my pen works, thank you. Must be the way I hold my tongue or something. If you're crossing two doubly heterozygous individuals together, I'm not sure that's any more legible, you're going to get that what you see on the right-hand side. Now, the hypothesis of dependent assortment was disproven. So this supports Mendel's idea that these alleles go off and form gametes all on their own. Wherever the yellow allele goes, what gamete it goes into, what cell, okay, that's being produced during meiosis, is independent of where the ones for seed shape go. They independently assort. So that's why you have to figure out all the possibilities. And if you wanted to know how many of them are going to be dominant for the first trait, like they're going to be, what, yellow, and then recessive, wrinkled, well, that's going to be 3/16 of them. It's a 9-3-3-1 ratio, and that 9-3-3-1 ratio, which is in red, the 9/16 of them will be dominant for both traits. 3/16 will be dominant for one and recessive for the other. There's two variants of that, the green round and the wrinkled yellow, and then 1/16 will be recessive for both. Now, you can also do a dihybrid test cross, but it's basically just two monohybrid test crosses. So, practice doing a dihybrid test cross. Take something like this. Practice this before you come to lab, too. So, again, you have some experience with it. And you want to test it whether it's true breeding or not, okay? So, you go out into the garden and you're going to pick, just like you did with the white flower, you're going to pick a green and wrinkled seed. Why would you pick another yellow or round seed of any variant? Because then you introduce another variable. Run that through a Punnett square, and then on Tuesday, be prepared to tell me, what the ratio, you would expect the ratio to be from something like that if this thing was not true breeding. If it is true breeding, that's easy. They'll all be yellow and round. If it's big Y, big Y, big R, big R. Oh, Penny, I could, but it would take me forever. I mean, this is the square right here. I would love to, honey. I'm sorry. It's just it would take me forever with my pen not working the way it is. But that's what I mean by a Punnett square. And the other day in class when I was making those, I was just drawing that out and writing the column and the row headings, okay? Does that make sense? If you want to stick around after, I can try to draw one. But again, it's a little more difficult to do. I might be able to do something on on the whiteboard though for you. You practice out drawing it yourselves too, right? Like once you get the gametes, right? Once you figure out what goes in the rows and the columns, and you get that by foiling, and that's the part I did do on the board there, right? The first alleles can make a gamete circle though, so you don't forget that they're gametes, they're sperm or egg. throw them in a table, then the rest is just copying it down. Hi, Dorian. How are you? Now, there's also those traits that we talked about that are a little bit different, right? There are for every rule there is in science, there's always a curveball. That's like in bio too, why I'm always so, if someone says, are there any organisms of this type that are this way? I'm like, I got to think about that. Like somebody said the other day, are there any photosynthetic animals? I'm pretty certain there aren't any of those, but it's always one of those things that we discover in science. Some of the early work that was done, as I mentioned, was done with plants. And so these particular plants show a type of dominance that's a little different than that complete dominance we've been seeing with the purple and the white. What I mean by this is the red allele, and again, however they abbreviate alleles, it doesn't matter, as long as it's defined. So right here, that's representing the red allele. The red allele is incompletely dominant over white. So what we mean by that is we get something called an intermediate phenotype. Isn't that a cool word for that? As an intermediate phenotype. So red and white, you mix them together and you get pink, don't you? Which goes back to the idea that, oh, maybe inheritance is blending. Maybe inheritance is blending things together. But you realize that's not the case. It's not blending. As I gave you the example of a tall parent and a short parent, like my dad was quite tall, my mother was quite short. So I sort of fit that model. But if you think about those kids out there that are taller than both of their parents, you can't mix red and white and get something that's more red. It's going to be pink. Now, this does not mean that the trait, this does not mean that the alleles became pink alleles, because notice what happens in the F2s. More specifically, notice what the phenotypic ratio is. The phenotypic ratio was 1 red one, 2 pink ones, 1 white one. 1, 2, 1. It's actually the genotypic ratio, but because the intermediate ones, these guys right here, and then the pen doesn't work. These guys right here, they fit that intermediate phenotype, okay? Does this happen in all flowers and all traits? No, just in some. And when it does, we call it incomplete dominance. I give you this eye color chart. I'm not going to have you do problems or memorizing this stuff, but people are always asking me about that. Eye color is actually inherited by more than one gene. And so there are several combinations. It's 16 possible combinations of color, and I think it may be even a little bit more complicated than this. But you can see, you can get an idea where hazel and green and all those different eye colors come from, right? Brown is dominant, okay, but with 2 different traits, like that one in the lower right. So sea slug, the algae. Okay. Yeah, that's good. Yeah, and it's kind of like I think I mentioned no, it's in Bio 2 yesterday. Same thing goes on in corals. Corals are an animal that actually have a photosynthetic protozoan living inside of them that photosynthesizes. Yeah, but that's not, why would you want one, Jordan? But that's not the critter actually photosynthesizing, that's the algae. But yeah, that's a good example. It's what we call a symbiotic relationship. Why would you want a sea slug? They're very cool. Look them up online. They're gorgeous critters. I saw one diving in the Philippines. It was just amazing. Yeah, slugs are neat. Slugs are snails without shells. Okay, as you can probably imagine it in life, as I talked about, things aren't simple as just 2 alleles for all traits. There are some traits out there that have multiple alleles, and human blood typing is one of those. And again, don't get too caught up. The capital I, other than it means it's dominant, the I think, stands for immunoglobulin, which is a protein in your blood. So A is one of the alleles, B is one of the alleles, and they're both dominant, notice. So now we don't have that, incomplete dominance. We have codominance, and then these guys here, that's recessive. Right, that's the recessive. That's the O allele. That's why we call this ABO blood typing. So you can be A, right, homozygous or heterozygous. You can be B, homozygous, heterozygous. If you're heterozygous for the dominant alleles, then you are heterozygous phenotypically. You're going to be AB. And then if you're that way, you're going to be O. Now, this is another time for me to remind you that, or let you know that just because recessive alleles doesn't mean it's the least in a population. For example, I think it's estimated among white males of Caucasian descent, just as an ethnic lineage, about half of them have O blood. Half of them have homozygous recessive blood. Up above, if you're wondering what this thing is in the upper right, if you ever get in, especially in A&P, and you talk about universal donors and acceptors, and I always got those backwards, but this helps me remember. People with O blood are universal donors. If you do a blood transfusion and you're A blood and somebody gives you B blood, it clumps up and it kills you. It can't happen. So O can donate to anybody and AB can accept from anybody. So O can donate to A's and B's. And if you want to know why, your book explains it to you. But that's, yeah, I think that's a neat little chart. I found that the other day, or a few classes ago, a few semesters ago. This next slide, or actually next two slides, shows you how powerful genes can be. For example, an example of pleiotropy is if you have one gene. Pleiotropy is defined as one gene that controls many traits, many phenotypes, phenotypic traits. Phenotypic traits, again, remember, don't have to be good. Heart failure, pneumonia, kidney failure, rheumatism, ain't none of that good. But those are traits, okay? So sickle cell anemia, those are the traits. So pleiotropy is one gene, many traits. Epistatic is an example of a pleiotropic gene, right? Pleiotropic, in this case, the epistatic gene is the E allele, the E gene, sorry, not the E gene. And if you're homozygous recessive, a lot of these things like this, like albinism in human beings, for example, is an epistatic gene. Albinism, being this way, homozygous dominant means you're not an albino and you're not a carrier for it. Someone like this would not be an albino, but they would be a carrier. And then a person like this would be an albino. Albinism is inherited a relatively simple way like that. Right, so the way you get two normal skinned folks, and this has been documented, two folks of African American ethnicity, so two black folks have actually had albino children because they're both carriers for this particular trait. That's the only way that could happen. But that albino gene, skin color, as we'll talk about in the next slide, is inherited by a lot of different genes. But that albino gene, in this case, or epistatic genes, remind me, as I mentioned in class, of multiplying some big mathematical thing by 0. If you look at some big mathematical formula and the whole thing's multiplied by 0, anything multiplied by 0 is 0. So to me, if you're homozygous recessive for the epistatic gene, like the golden labs are down in the lower right, see two little e's, it doesn't matter what B says. See, B is controlling the color. Homozygous dominant, you're going to be a black lab. Heterozygous, you're a black lab. Homozygous recessive, you're a brown lab. This guy right here, come on, Pen. He's homozygous recessive. But notice all those guys are either homozygous or heterozygous for the E. But then when you're E, it doesn't matter what this says. You're going to be a golden lab. So there are genes like that. There's even one, what I mentioned in class, called testicular feminism. It's a gene that can be inherited. It's relatively rare, obviously, in which, let me see, get this right. The individual is an XY, which is a male, chromosome-wise, but phenotypically is a female. And it's actually, it's not hermaphroditism or anything like that. It's actually, I think there's a case, if I recall, years ago of a woman, I think I told you guys in class, of a woman from one of beauty contests, and then later on couldn't get pregnant, and for whatever reason they did some tests on her and found out that she indeed was this. She was actually a male, but for reasons that are beyond the scope of this course, your body can do some very interesting things with genetically, and especially with epistatic genes. Many traits are what we call polygenic. All right, human height. So this is a bunch of soldiers standing. There's some a few short folks, a few tall folks. Most of us are kind of in the middle. And that's why the reason behind that is because what we mean by polygenic inheritance is the opposite of pleiotropy. So this is many genes, polygenic, controlling one trait. And many genes, by the way, and those genes can have many alleles. So just to make something kind of simple, looking at this without kind of, just overwhelming you, we're talking about a trait that has 3 genes, A, B, and C. And then we're talking about gene A has got 2 alleles, a big A and a little a. Gene B has got 2 alleles, a big one and a little one. Gene C's got 2 alleles, a big one and a little one. Now, they didn't put it in this chart, but let me remind you, you could also have a gene B could have 12 alleles, and gene C could have 8 alleles, and gene A could have 4. They could all have multiple alleles like blood typing. Now, the way you're only going to have 2 alleles for the trait because you only had 2 parents. So you're only going to have 2 A's, okay, either a big one or a little one. So if there were 12 alleles, you might have, I got to switch the numbers if you let me, you might have allele 2 and 2 or 2 and 4. You know what I'm saying? There's a lot. It's kind of like I was saying why there's so many different breeds of dogs, so many switches. So that's why when you look at that chart, and don't worry about the numbers so much, but look what they're doing. When it comes to alleles for skin color, there's everything in between. There's very light skin, so you're recessive for everything, down to the, where's it, the lower right? Yeah, lower right, where you're dominant for everything, okay? So that's how most traits work, and that's why we get that. Hair color would fit into that. Lots of other traits. There are some traits that are simply one gene controlling one trait, but for the most part, in organisms, we're talking about genes all kind of working together because it's variation, folks. You need variation. Variation is important for reasons we've talked about before. Now, I get a question, or maybe you've heard before, the nurture versus nature. Right? Nurture versus nature. Is that kid a rotten kid because of the way they were grown up or raised, or because of their genetics? And I have 2 kids, and I'll say they were raised similar, right? And they're very different. So there is a nature and nurture kind of thing to that. A really good example is my wife loves hydrangeas. Those are hydrangeas, those flowers. And these are all the same species of hydrangeas, same species, but different colors. And that has to do with the soil they grow in. So that's the title of this is, yeah, absolutely. Right? It could be a little bit of, how you were raised versus your genetics, My son is a type B personality guy, and it's got to be the way he was born, because I always said he's basically got the metabolism of a reptile. He's not slow in any way like that, but he's just very kind of lay back and that sort of stuff. So the environment, maybe. These are what we call multifactorial traits, genes plus the environment. What the hydrangeas there, the gene for flower color is affected by the pH of the soil. So if you want to have, is it blue? If you want to have blue hydrangeas, you put coffee grounds. Coffee is acidic. So I think that's right. So if you want blue hydrangeas, you want a low pH for the soil. And if you want red hydrangeas, you want more basic. And if you walk out and look at a hydrangea bush, my wife's got some pretty good size hydrangeas out there. Sometimes the colors are mixed in. And I'm like, oh, what does this mean? It means minor variations of the pH of the soil. So flowers that are being supplied by one roots that are growing in a soil that might have a little bit different. I didn't realize the doggone things were so sensitive. But that's why you sometimes get variants of colors. So there is obviously a relationship, isn't there? Courtney, I lied. We may not get to 13 today. I'm kind of having fun with this. Doesn't matter. We'll get through it all. So if you want to do that and maybe find a flower area in your flower garden, plant a row of hydrangeas. The longer the better, right? A really long row, and see if you get a bunch of different colors. And you could even experiment. You could use them like oh my God, you could use them like a pH scale. I wonder what they look like in Nutri. They're probably so sensitive, it'd be hard to keep the pH they're probably hard to keep the pH normal. Okay, pedigree analysis. Pedigree. So this is, squares are males, round ones are females, and it varies, but they're always a key. In this particular case, if they're colored in purple, it's a male who has the trait, they're affected. It may not be a good trait or bad trait, it's just a trait, okay, and then affected females are round. Mating is indicated by the square and the round, right? Then the offspring are indicated by however many you have born, right? Everybody's seen these, you understand how this works. What I want to do here is see how we can use a pedigree analysis to answer a question about this thing called widow's peak. Is widow's peak dominant or recessive? Now, don't answer the question, but if you know, and I think I actually gave it in class, but see the woman on the left, she's got the widow's peak. The woman on the right does not. So we're trying to figure out, you know, how that came about, how that was inherited. So the woman on the left is represented obviously by a circle since she's a woman, and since she's affected, we're going to color in purple. Her sister, as you can see, that's obviously her sister, is not affected. Both of her parents were affected. And then, let's see, if we go back to her dad's side, then the grandfather, he had a widow's peak. One of her aunts had a widow's peak. All right, and then on the mother's side, what do we got? The grandmom had a widow's peak, but one of the aunts did not. Both brothers did not. Whew. Now, doing pedigree analysis can get kind of confusing. All right, you take a look at it, and you're like, how do I know? Well, focus your attention right here. Focus your attention right there. Okay? Already gave away. I don't want you to give away the whole answer. Focus your attention right there. All right. If widow's peak is dominant, let's go with that first, right? That's our first hypothesis. Is if widow's peak is dominant, let's take the individual on the left, okay, and take the individual on the right. Now I'm writing dominant, but I'm only writing what I'm knowing. Okay. If it is dominant, all right. If it is dominant, is it possible that those two people that mated right there could produce individuals that both have a widow's peak and do not? Yeah. Yeah. Let's ask the other question then. What if widow's peak is not dominant? If widow's peak is not dominant, then those two parents are homozygous recessive. So we got two choices. Is it that? Or is it a recessive trait? So those two parents that are purple, right? These two parents right there. I said, will you focus? If widow's peak is a recessive trait, could they have produced this female? Well, if widow's peak is a recessive trait, right, could they have produced this female? The problem is they couldn't have produced one of both, right? So if we take a look at it over here, two dominants could produce a recessive, but two recessives cannot make a dominant. So if widow's peak was recessive as a phenotype, if the purple parents were both this way, there's no way you're going to get this girl right here. Like you all were saying, right? Someone went yes, someone went no. But no, you're not going to get this offspring. If widow's peak is a recessive trait, you cannot get a dominant offspring. So the trick to doing pedigrees and answering these questions is finding when two affected parents mate. Finding the two purple, the one purple square and the purple, when those mate, look at the offspring. Now you can answer the question. Now, if they only had one offspring, well, that's the problem. But if they got more than one offspring, if the trait is recessive, none of their offspring can have the dominant allele. It's kind of like I said in class the other day, two blue-eyed parents. Now, if we stick with just a much simpler idea of eye color, and this fits in with the chart I showed you earlier, but two blue-eyed parents could never have a brown-eyed child. Two white Mendelian flowers, could they ever have a purple one? Could two white flowers ever have a purple one? No, never. All right. So what's the key to figuring out whether the trait is dominant or recessive? Find the two parents that are affected. if those two parents produce any of the individuals who are dominant, if they produce any unaffected, then that trait obviously has to be dominant. Okay, this is the key right here. Two dominants can make a recessive. Two recessives cannot make a dominant. Two recessive parents can never make a dominant offspring child. Now, again, did I put all the letters? There they are. So there we go. Ask her the question. And there's our answer. The only way that we're going to get this child over here on the right is if this trait is dominant. So widow's peak is dominant. All right, just play devil's advocate, right? Put two little W's in both those parents. You're never going to get that child on the right. Now, as I said, most genetic disorders, most inherited genetic disorders, like albinism, for example, testicular feminism, most of those are recessive. And the reason why that is because evolutionarily speaking, those individuals many times would be, the word I use in bio too, it's called least fit. For example, if we look at albinism, right? Albinism, AA, 2 capital A, normal, 2 carriers, it's a 1-2 to 1, a 3-to-4 ratio, just like Mendel. So two parents who are carriers for albinism have a 25% chance of having an albino child like that young girl on the right. Albinism is not something that would be beneficial. Albinism means that you have a lack or you have a low amount of melanin in your skin. Melanin absorbs ultraviolet rays, what gives you a tan. The reason why you get a tan when you lay out in the sun is ultraviolet rays ain't good for you. Your body's trying to filter them out. It doesn't filter all of them out. Folks without melanin, or folks who have low levels of melanin, light-skinned folks, have a very difficult time in filtering out that UV. And if they can't filter out that UV, they suffer from higher rates of skin cancer, right? Skin cancer, or in other types of cancer, well, skin cancer predominantly. Albinism occurs in other things. It occurs in large other mammals. It occurs in plants. This is a fern that's an albino. Gators, we all know it does in that. It occurs in sharks. This is a white-spotted bamboo shark, and this is a albino shark that was born at SeaWorld. I actually have that one in the collection at school. I wrote a paper on that because that was the first time that species had an albino. Albino human beings. This is an albino from Penny and Penny in my time. Penny, do you know what that is? What's a reversed albino thing? That's Edgar and Johnny Winter. Edgar and Johnny Winter had a rock group when I was a kid, and it's called. There is some melanistic individuals, Jordan. Like I know among species of guppies, they'll have a shape, and then some will be a little darker than everybody else. And they're known as melanistic. Yeah, that's one of the brothers. They were not the albinos. They were identical twins, and so I think that was probably made part of the shtick, right? But albinism is not good for filtering out UV rays. And this white gator, man, if you're white, it's not very good at sneaking up on things. So albinism is a recessively inherited genetic disorder. Type O blood is recessive. It's not a disorder. But oftentimes the reason why genetic disorders are recessive is because those individuals aren't going to be as fit and produce as many offspring in the environment. Either you're dealing with skin cancer issues or this poor gator who can't hide. The only thing he could sneak up on would be a polar bear. Oh, who males in the room? Just Seth and I. Seth, you and me, we're nothing more than a female. Part of our chromosomes missing. Did you know that? That's what a Y is. You have brown eyes in both your parents. It depends to make sure, and that's where you got to look at that eye color chart. Remember, there's different shades of that, so it's not as set as brown and blue. Remember that eye color chart. It could be your parents may have. You said your parent, you have brown. Maybe you have hazel eyes, or your parents had brownish hazel eyes. Take a look at that chart. Yeah, a lot of times it has to do with what you're wearing. Your eye color has a lot to, well, your eye color has to do with how much pigment is deposited in the iris of your eye. That's the colored part. The more pigment, the darker the eye. So lighter eyed folks like myself, for example, and Courtney's dad, if you have lighter colored eyes, a lot of times the eye color comes. If I wear a blue shirt, the blue light, remember back in photosynthesis, I did the stop sign. The blue light coming off my shirt actually reflects through my eyes, makes my eyes look a little bluer. Wear a green shirt, looks a little greener. Did you get what I said, Seth? Did you get what I said about we're only part of a female chromosome? See, there's the X and the Y chromosome. The reason why the X and the Y chromosome can even pair up is the fact that they have homologous segments. They have areas that allow them to pair up together. Remember, the other ones are copies, you know, or identical. They're homologous. These don't look too homologous. But the part of the Y that's there is homologous with the part of the X, part of the X there. I think it's like this part down here. So we're only part of an X chromosome. Females have two X chromosomes. We have an X and a Y. The Y chromosome has something that's called an SRY gene, and that's a sex-determining region, right? That's what causes testes. So if you have two X's, you don't have that SRY gene. If you have an X and a Y, you have that SRY gene. And that SRY gene, even though it's only on one chromosome, is expressed, and you get testicles. You become a male. Testicular feminism, that SRY gene, an epistatic gene, blocks that. So even though you have the Y chromosome, you don't get testes development. You become females. Okay, very quickly through the sex system, if we're talking about human beings, we have 44 sets of autosomes. I talked about this in the karyotype. That last pair in human beings, you know, it happens to be 44, plus that last pair is known as our sex chromosomes. 44 or 22 pairs, right? And males produce an X or a Y sperm through meiosis, right? Yeah, each one of those autosomes has to end up in a gamete. Each one of those sex chromosomes ends up in a gamete. So back to ancient England and all that stuff, who determined the sex of the offspring? So the queen was supposed to give the king an heir, and yet all along it was him who was deciding it, wasn't it? Because females only have X chromosomes. See, in those and in insects down below, insects do a little bit different, a little bit simpler. Sperm determines the sex, because in the insects, the way you're females, you have two X chromosomes, and if the sperm doesn't bring in a sex chromosome at all, in other words, only the egg has an X, if you're an XO, then you're a male. So it's kind of like ours to the next degree. Instead of having only half a Y, they ain't got no Y at all. They have no other sex chromosome. Doesn't mean it's the same way in other organisms. Like, for example, in the chickens, it's the egg that determines the sex, right? See, the females are different. There's ZWs, and the males are ZZs. And then there's even another way. Among bees, it has to do with whether the egg is fertilized or not. Check that out. So males are haploid and females are diploid. So it's not quite the same as, you know, just XY, and there's all kinds of different ways of doing this. And then there are those that I mentioned you guys before, that there are no males. There are species out there that are no males at all, just females. It's called parthenogenesis. And it's not just in weird little critters. There's sharks and lizards, some fish that can engage in this. Okay, now, the reason why we have to know how sex is inherited is it answers a question. Does anybody know someone who's colorblind? It answers this question. Does anybody know anybody who's colorblind? Or has hemophilia? Or Duchenne muscular dystrophy? Duchenne is, whoops. Is it two E's? I can't remember. Nobody knows anybody who's colorblind? Ever heard of anybody who's colorblind? And what sex are they, Jordan? Male. Does anybody know someone who's female colorblind? Ah, your friend. Your friend, is her father colorblind? Dorian, your friend is she's female or male? I'm sorry. That what note means? Gotcha. We don't often find colorblind females. They do exist, but here's a rule. Here's another thing that you can have fun with if you want to over the holidays. Colorblind females have to have colorblind fathers. Now here, let me show you why that is. The gene for colorblindness is what we call an X-linked gene. It's not just colorblindness. There's Duchenne's muscular dystrophy that I mentioned, and hemophilia, you know, the one where you can bleed to death by bumping into the desk. It's not funny, but it's horrible. Folks with hemophilia have little to no clotting factors. All right, those traits, which have nothing to do with sex, are on the sex chromosomes. They're X-linked. They're on them. So in this particular example up here that you see, on the male on the right-hand side, the one up here, it's colored into effect that he's affected. He's colorblind. He's got the colorblind trait on that X chromosome. Now, colorblindness, Duchenne muscular dystrophy, hemophilia, those aren't good traits. So these are recessive disorders. All right, these are recessive disorders. So let's take a look at how this is inherited. So the male produces sperm, which are Y, and he produces X sperm that have the colorblind trait, whereas the female produces normal eggs. So what we end up getting is no colorblind children, but notice the females. They're not colorblind, but what are they? That's right, they're carriers. Notice the males, a colorblind male, as long as his wife is completely normal, a colorblind male, none of his sons will be colorblind, right? None of his sons will be colorblind because the Y comes from him, the X comes from the mother. It's the X we're worried about. So if the mother is a carrier, so let's go down here. If the mother is a carrier and she mates with a male who does not have colorblindness, you got a quarter chance that you can have a colorblind child, half a chance that one of your males would be that way, and a quarter of a chance that one of your children is going to be a carrier, and that's going to be the female. Now, if mom's a carrier and dad's colorblind, then you're going to get half of your children will be colorblind, and this, folks, is just a Punnett square. So the reason why we see these X-linked traits more often in males is because it's a recessive disorder. If you're a female, you have the chance to play dominance and recessive. So if I draw a C, and I could draw it in either X, it doesn't matter, say I draw it there. What I'm saying there is you have one dominant X and one recessive X as it comes to this trait. That's what the C means there. The C means you have the recessive disorder. But males, remember, if you have the recessive disorder, you have no other X to be dominant over it. See what I mean by that? So this is a recessive disorder, but that really only comes into play, this whole idea of being recessive, if you got two of them, and you got to be female to be that. You got to have two X's. Now, both those X's could be clean. One of those X's, you could be a carrier, or if you're colorblind, and then let's say your mate is colorblind, the whole family is going to be colorblind, right? You got to have a good X out there somewhere. That's why the family on the right there can produce some children who aren't colorblind. This female is not. This male is not. She's a carrier. This female is colorblind. See, colorblind daughters got to have colorblind fathers. Works the same with hemophilia or Duchenne. Hemophilia, if you were to just to change the C to an H, you'd be doing the same thing. There's some of these X-linked genetics problems in those examples too. All right, the last thing we're going to talk about here is something known as X inactivation. It has to do with something you might have heard about called a Barr body. In the female cats, all right? So I've got to stress female cats because you're going to have two X chromosomes. Look up at the top. You got the allele for orange fur and you got the allele for black fur in this particular cat. So we have a heterozygous cat. Now, what happens in these organisms is they have two X chromosomes, and throughout their somatic cells, one of those X chromosomes is going to take a back seat. All right, one of those X chromosomes is going to become inactivated all throughout those somatic cells. So we're not in meiosis, but this doesn't work to happen in males. Okay, in males you can't have this because we don't have another X chromosome. You inactivate the X chromosome in the cell, it's the only one they got. Females got two two X chromosomes. So randomly throughout the cells, the somatic cells, the diploid cells, one of those X's inactivates. It becomes the Barr body, named after some guy who first saw them underneath a cell. So now that's random where that's going to happen. That's why the old question, it's kind of like colorblind males, why are most calico cats female? Now, do you know why that is? Most, I said most, not all. We'll talk a little bit later on in the next chapter about why not all of them are. But why are most calico cats female then? Have you ever heard that before? Cats with this coloration? Or tortoiseshell. Am I saying calico? Maybe, wait, no, it's calico. Has anybody heard that before, that calico cats are female? Mostly. Hello, I haven't lost everybody, have I? Why? Why? Oh, this is on. Here we go. Okay, if you look at calico cats, why are most calico cats female? The reason why they're female is because they have two X chromosomes, and two X chromosomes makes you female. Now, okay, I'll go ahead, last minute here. How is there a male calico cat? There is something that happens, and we'll talk about later, in some males who have two X's and a Y. Now, as soon as you have a Y, you're a male, no matter what else you got. This is a disorder, but sometimes a male cat, XXY, will be a calico because you got the two X's there again, and one becomes active. Okay? All right, any questions on any of that, folks? That's it for today. That's all for the lecture. We'll do chapter 13 because that is the last slide in this chapter 12. Yep, we'll do chapter 13 next week on Tuesday. Work on those genetics problems. Please don't come to class without having those at least tried out, just so it, I got an idea of, where the level of knowledge is and what I need to work on. The monohybrid ones are easy. Remember, you get an allele from each parent. If I'm asking you to calculate numbers, think about those phenotypic ratios and those genotypic ratios that I've given you, okay? All right, let me turn the recording off.